Hello, we shall be discussing on uses of microbes in sewage treatment. Sewage is the municipal wastewater generated in the cities or town and may contain human and animal excreta or other organic waste, and may also contain a large number of pathogenic microbes such as bacteria, fungi, protozoa, etc. Let's discuss on the sewage treatment. Sewage treatment is carried out in the sewage treatment plant, and it involves two stages. The first is the primary stage and the second is the secondary stage. In primary treatment, it involves physical process of removal of large and small particles from sewage through sequential filtration and sedimentation. Here, the sewage is passed through the screen to remove large floating objects, and the filtered sewage is then passed into another chamber where coarse solid matter like sand, small pebbles, etc. are allowed to settle under the influence of gravity. After this, the sewage is passed into a large primary settling tank with a gentle slope, and the solid material and screen organic matter constitute the primary sludge. Primary sludge can be used for preparation of compost or manure, or it can also be directly used for generation of biogas. The effluent that is produced from the primary treatment after the removal of primary sludge is then passed into another tank where secondary treatment is carried out. Secondary treatment is also called as biological treatment. The secondary treatment involves biological process of microbial degradation of organic matter. There are several methods for secondary treatment such as oxidation tank, trickling, filter, and activated sludge. However, we shall be discussing on the activated sludge. In the activated sludge method, the primary effluent that is obtained after the primary treatment is passed into a large aeration tank where it is constantly agitated mechanically and air is pumped from outside. A large number of aerobic microbes such as algae, filamentous fungi, bacteria, and protozoa are allowed to grow in this secondary biological treatment. So these microbes, after growing, they form a mass called as floc. These microbes that is constituting the flocs consume major part of organic matter in the effluent, and they reduce the biological oxygen demand, this biological oxygen demand considerably in this sewage. Now the effluent is passed through the sedimentation tank where microbial flocs are allowed to settle down. This settled material is called as the activated sludge. The major part of the sludge is then pumped into a large tank called as the anaerobic sludge digester. This tank, in this tank, anaerobic microbes are allowed to grow, and these anaerobic microbes start digesting the bacteria and fungi that is present in the sludge. Now, during the anaerobic digestion, bacteria begin to produce a mixture of gases, that is methane, carbon dioxide, and hydrogen sulfide. So this mixture of gases is highly inflammable and can be used as a biogas. Now the spent sludge after the secondary or the biological treatment can also be used as manure or as compost. The remaining portion of the sewage can be directly released into the natural water bodies like rivers and streams. Now we shall be discussing on the uses of microbes in antibiotics production. What is antibiotics? Antibiotics are drugs that are produced from microorganisms which in low concentration may kill or even retard the growth of the disease-causing bacteria. Some of the bacteria that produces antibiotics include Bacillus subtilis and Streptomyces graeus, and among fungi, Penicillium cephalosporin and Aspergillus are used in antibiotics production. Antibiotics function as bactericides or bacteriostatic. Bactericides here refers to ability to kill bacteria, and bacteriostatics here refers to inhibiting the growth of the bacteria. So the antibiotics may be both broad spectrum or specific. Broad spectrum antibiotics can kill a large number of pathogens that belongs to different groups with different structure and cell wall composition. However, a specific antibiotics is an antibiotics which is effective only against certain type or one type of pathogens. Therefore, an ideal antibiotics should have following characteristics. One is that it should be harmless to host without side effect. Two is that it should be harmless to normal microflora of alimentary canal. And three is that it should be broad spectrum, that is effective to varieties of pathogens. And the fourth one is that it should have quick action. Bulk of antibiotics are produced from three groups of microorganisms. The first one is eubacteria or true bacteria. The second is actinomycetes, and the third is the fungi. Some of the antibiotics that are produced from bacteria are bacitracin, gramicidin, poliomyxin B, etc. And some of the antibiotics that are produced from actinomycetes involve chloramphenicol, erythromycin, neomycin, etc. And some of the antibiotics that are produced from these fungi include penicillin, cephalosporium, fumigillin. And the first antibiotics that is produced was penicillin, and it was first produced from the mold named Penicillium notatum by Alexander Fleming.